Welcome to Let's Learn Plants. Today, in this explainer, we're jumping straight into a fascinating, invisible battle happening right under our noses, or, well, right above our heads, in the leaves of literally every tree and plant out there. We're looking at how plants protect themselves from intense sunlight. You might think plants just sit around passively soaking up the sun's rays, but dealing with all that energy, it actually requires a highly sophisticated microscopic security system. So let's get right into it. Here's our quick roadmap for today. We'll be looking at 1. The threat of photoinhibition, 2. Carotenoids as the protective shield, 3. The Xanthophyll Cycle volume knob, and finally, defense systems and D1 repair. Okay, let's dive into this first section, the threat of photoinhibition, and really understanding the danger of excess light.
It sounds kind of like an oxymoron, right? Plants literally need light to survive. It's the entire engine of photosynthesis. But just like a high-performance car engine can easily overheat, too much light is actually a deadly threat. And this brings us to the core biological conflict the plant has to solve: photoinhibition. This is the condition where photosynthetic organisms are actively damaged by excess light energy. It happens because the plant's pigments absorb way more energy from the sun than they can possibly store through photochemistry. Imagine trying to catch 100 baseballs thrown at you all at exactly the same time. You just can't do it. The photosynthetic membrane is incredibly delicate, and if that massive influx of energy isn't managed, the whole system breaks down.
And that's exactly why plants have evolved mechanisms to deal with this overload. Our source material puts it perfectly with this quote: "Photoprotection acts as a safety valve." I really want you to keep this visual in your mind as we go through the rest of the explainer. The plant isn't just sitting there taking the heat, it's actively dissipating this excess, harmful energy, venting it out safely before the pressure builds up and destroys the delicate cellular machinery inside the leaf. Moving on to section 2, carotenoids, the protective shield and the plant's first line of defense.
So what exactly is acting as this biological safety valve? Well, to really appreciate why carotenoids are so heroic here, we have to look at the absolute worst-case scenario. Consider a normal, safely quenched chlorophyll molecule, where energy transfer happens smoothly. Beautiful, right? But contrast that with the danger of unquenched chlorophyll. If an excited chlorophyll molecule isn't quickly relieved of its excess energy, it'll react with ambient molecular oxygen, and this creates something called singlet oxygen. Singlet oxygen is highly reactive and incredibly destructive. It causes severe oxidative damage to the lipids that make up the thylakoid membranes in the chloroplast. It is essentially a microscopic wrecking ball let loose inside the cell.
Answer: the carotenoids. These are essential accessory pigments that courageously step right in front of that wrecking ball. Carotenoids prevent this catastrophic damage by simply accepting that excess energy directly from the excited chlorophyll. They take the hit like a shield, and then they return to their normal ground state by releasing that energy as completely harmless heat. And the true genius of biology here? The excited state of a carotenoid physically doesn't have enough energy to form that dangerous singlet oxygen, so the threat is totally neutralized. No singlet oxygen, no wrecking ball. This brings us to section 3, the Xanthophyll Cycle volume knob, also known as Non-photochemical Quenching, or NPQ for short. We all know a plant's environment isn't static. A dark cloud might pass over, or the sun might suddenly hit intense high noon. Plants dynamically manage these changes through a process called non-photochemical quenching, or NPQ, and this brilliantly illustrates the plant's incredible adaptability. The text describes NPQ as acting like a volume knob that adjusts the flow of light excitations reaching the Photosystem II reaction center. It keeps things at a manageable level. As the sunlight gets brighter and more intense, the plant simply turns that protective volume up, safely dissipating more and more of that excess light energy as heat to keep its biology perfectly balanced. So how does this volume knob actually work? The mechanics are driven by a specific group of carotenoids called xanthophylls, operating in what's known as the Xanthophyll Cycle. It involves the constant interconversion of 3 specific molecules: violaxanthin, antheraxanthin, and zeaxanthin. When a plant is suddenly exposed to high light conditions, an enzyme called violaxanthin deoxidase springs into action. It actively drives the conversion of violaxanthin all the way into zeaxanthin, using antheraxanthin as a quick intermediate step. Basically, the greater the light stress, the more zeaxanthin the plant pumps out to protect itself. Why does the zeaxanthin matter so much here? Because it physically alters the plant's light harvesting structures. When zeaxanthin binds together with protons and specific light harvesting antenna proteins, like the PSBS protein, it causes conformational changes. Now that's just a fancy scientific way of saying it physically changes the shape of the proteins.
This new shape strongly promotes the rapid quenching of chlorophyll, venting out the dangerous energy as heat. The plant is literally reshaping its own microscopic anatomy in real time just to survive the midday sun. Which leads us into our final section, Section 4, Defense Systems and D1 Repair. What happens when those primary defenses fail? Because let's face it, nature is relentless. Sometimes the sun is just too intensely focused for too long. What happens when these primary quenching defenses are entirely overcome? Even with the volume knob turned all the way up to 10, extreme light stress can still overwhelm the plant, leading to photoinhibition that actively damages the Photosystem II reaction center. When this inevitable damage occurs, the plant relies on a highly structured, multi-level defense system. First, as we've discussed, is quenching, dissipating the heat. Second, if toxic photoproducts do happen to form, the plant deploys scavenging systems to hunt down and eliminate those dangerous reactive molecules. But if the damage persists and the physical machinery actually breaks, the plant enters phase 3, active repair, specifically targeting a crucial component called the D1 protein. So the crucial point is understanding how this final, dramatic failsafe actually works. How exactly does the plant repair a completely damaged Photosystem II complex when its core machinery has been blasted and broken by too much solar radiation? Well, the plant executes what's basically an incredibly efficient cellular pit stop. The main casualty of photoinhibition is almost always the D1 protein, which is located deep within the reaction center. But instead of throwing away the entire massive Photosystem II complex, which would be a terrible waste of metabolic resources, the plant carefully extracts just the damaged D1 protein from the membrane. It then rapidly synthesizes a brand new D1 protein, inserts it perfectly into the gap, and restores the function of the whole center. The plant scrambles to swap out just the broken part, meticulously recycling and saving the rest of the undamaged machinery, so it can get right back to work immediately. It's an absolutely astounding feat of biological engineering, and it's happening millions of times a second in a single leaf on a sunny day. Which leaves us with this final, provocative thought to ponder. What would happen to the entire foundation of Earth's food chain if plants couldn't constantly execute this microscopic repair? If they couldn't quickly swap out their D1 proteins under the intense summer sun? Without this tiny, invisible defense mechanism working tirelessly around the globe, green life as we know it simply wouldn't exist. I hope this explainer gave you a whole new appreciation for the secret lives of plants. Stay curious, keep exploring, and we'll see you for our next deep dive.